Hi, this is the lecture about the experiment performed by Matthew Meselson and Franklin Stahl. And this experiment was done in 1958 to confirm the model of DNA replication, that which one of the three model that was given and which of the three model was the true one. So Matthew and Meselson, in their experiment, we can see the first step. The concept was that the DNA contains nitrogenous bases. So he, both of them, they made a medium, culture medium, containing heavy nitrogen, 15N nitrogen, and they grew E. coli cells in those culture medium. So when cells are centrifuged, the DNA will show up heavy nitrogen. So all the DNA in that culture cells, E. coli cells, their DNA was supposed to found only the nitrogen that was present in the medium. It was 15N nitrogen. So he saw this kind of observation in this step one of their experiment. Now, experiment of Meselson and Stahl experiment, step 2. So in step 2, they transferred E. coli cells from the first medium that contain 15N nitrogen to the second medium that contain light nitrogen, that is 14N nitrogen. Now cells were allowed one round of replication only. In cells, growth takes place, and it was a very restricted growth, that cells were allowed to grow at only one cycle. And they centrifuged the cells, and what they found, so you can predict that the centrifuge tube would show each model like the semi-conservative or conservative or dispersive. So we can verify which one did he see. So if it is conservative, then there can be two band of DNA. One can be 14 N nitrogen, and another can be the 15N nitrogen. Like parent cell and daughter cell DNA can be the different one. So he could not find two bands in this second step of the experiment, but he found single bands. So which could be like a 15 mixture of a 15N and 14N nitrogen, or it can be like dispersive type. So he further again allowed to grow the second round of replication in the light nitrogen medium. So this is the second step. Third step. In the third step, the cells were allowed to grow for second round of replication, and after that again they centrifuged the culture medium, and they isolated the cells and extracted the DNA. So again, now predict what the centrifuge tube would show with each model. So it would be like this way. Conservative cannot be possible because it was rejected in second step. So in third step, he found two band in semi-conservative type, and if it is of dispersive model, then it can be only single band, because all DNA can be of like segments of old parental DNA and the new daughter DNA. So if it is dispersive type, then there can be only single band of DNA in the centrifuge tube. But he found two bands of DNA. One is 14N strand, and another is 15N 14N strand. So in the observation of third step, we saw that semi-conservative model was true, and conservative was rejected in the second step, and the dispersive model was rejected in the third step model of the Meselson and Stahl experiment. So this is what we can summarize, that first Meselson and Stahl started their experiment by growing the bacterial culture in medium containing 15N nitrogen, and after the bacterial growth has taken place, they transferred few cells from old medium to the new medium, and the new medium was containing 14N nitrogen. So in the second experiment, when he performed the second experiment, that then they did the restricted growth of the cells. So in the first replication of the cells, he disproves or rejected the conservative model of DNA replication, and in the second replication of the cells, they further rejected the dispersive model of the DNA replication, because they found only semi-conservative model type of DNA replication, which they were supposed to get two bands of DNA, one mixed strand like one parental strand, and another can be like new daughter DNA strand, and the second DNA strand second band was containing pure 14 nitrogen and containing the DNA in parental as well as daughter DNA. So this Meselson and Stahl experiment was the experiment to prove that the DNA replication takes place in the cells, in each and every cells of our body, only through semi-conservative model of DNA, not conservative, not dispersive model of DNA replication. Thank you.